Thanks everyone for being here. And so feel free to interact during the talk as well if something is unclear, or I'm happy to respond to short questions during the talk, given that it's maybe a subject that is not super common here at the ESL. So that's why I dedicated a first part about the context and kind of introduction about the gut microbiome and why I'm studying that, why is it interesting for me, but also for a broader audience. And the talk's going to be, it's relatively simply structured. It's going to be, so A, an intro, B, some pattern about the specificity of the gut microbiome. I'll talk about that later. And then some mechanism that can explain what we observe. So, I mean, have you heard about the gut microbiome? Or is it totally new to you guys? The kind of vague idea of what it looks like. And so,
oh no, it's no longer working. Try again.
It was working.
That's why you do testing, so that it doesn't work in the end.
Interesting.
So nothing is working anymore?
Escape.
We're back to get it.
Okay, cool. So if you want to study the gut microbiome, I guess the best way to start is to dissect an animal and look where that ecosystem is. And here you have a dissection of a rabbit, and you can see the digestive system starting here, the mouth all the way to the anus here, with the stomach, then the small intestine, the large intestine, and the rectum here. And if we zoom in to see where the gut microbiome is, you need to zoom in. Like let's start with the large intestine, one part here, and you zoom on the host side here, where you have an epithelium, so a contact between the lumen, so where the food is, basically, processed food is, and the host here. And you can see that there is basically a lot of host cells here. We don't see the microbiome yet. And if you zoom in again to that border here, what you see is in blue the host cells, so the epithelium, in green the mucus that kind of coat the intestine, and here with various color you see the microbiome. So you see a lot of different cells with different color that represent diversity of the microbes, so genetic diversity of the microbes. And so what you can see that basically the epithelium is coated with a microbial ecosystem that is abundant. There's a lot of microbes in your gut. There's actually like billions of cells. There is actually about the same number of microbial cells in your gut that eukaryotic cell. Okay, so you're kind of half microbe and half a human, if I may say. And so an abundant ecosystem and also a diverse ecosystem. So there is a lot of diversity in here. There is hundreds to thousands of species of microbes living in the gut of every mammal. So I'm going to talk about species for bacteria. We can discuss later if it's valid or not, but for the sake of clarity, I'll talk about species, bacterial species. So that's where this microbiota is, so coating the gut here of a rabbit, but also of human. And you may wonder why do we study the gut microbiome? Why is it an interesting ecosystem? Well, it's interesting, at least for one reason, is that it provides benefits to the host. So in a sense, it provides ecosystem services to its host, because it's really an ecosystem that provides benefits. And how do we know that? It's from actually a very interesting and pretty elegant experiment where you actually grow animals without a microbiome. So we call them germ-free animals. And so you grow them in sterile chambers, as you can see here on the bottom. And then you have this germ-free animal that are alive, right? So even without microbiome, you still live. It's not an essential organ. But by making comparison between the germ-free animal and the normally colonized animal in blue here, so between the yellow and the blue animal, you can actually see whether there is an effect of the microbiome on host fitness and host health in general. And when you compare these two types of animal, what you see that one is making more fat out of his diet. So basically, it's processing better the diet, which is fairly known, at least in cows, for example. We kind of all know that in the rumen there is microbes that actually process the grass for the cow and enable the cow to actually get the nutrient out of and the energy out of the grass. And so this happened because the microbe have the enzyme that are able to digest the carbs that the animal is eating, while the animal itself doesn't have this enzyme in its genome. So it rely on the microbe to digest those kind of hard to digest polymers. So that's why there is this beneficial effect on digestion, especially in herbivores. But it's hard to digest cellulose. And so that's one benefit of the gut microbiome, probably the most well-known benefit. But there is another one that is actually very interesting, and it's about resistance to infection. And so if you basically try to infect these two animals with a pathogen, the germ-free animal is way more susceptible to infection. And that's for two main reasons. The most obvious one for an ecologist, at least, is that it's for infection of the gut. And the gut microbiome, when it's already here, it kind of enter in competition with potential pathogen and kind of exclude them competitively from the gut and protect the animal from gut disease. That's one mechanism of protection. And another one, which is pretty interesting, comes from the fact that infections that are not located in the gut are also not as important in colonized animals. So for example, a germ-free rodent in that case are way more susceptible to a lung infection. It's kind of weird because the gut microbiome are not living in the lung. And so this is because of another mechanism. This is because the gut microbiome is very important to train the immune system to react properly to pathogens, especially during the first year of life. And so if you don't have that microbiome during this immune system development, your immune system is kind of messed up and tend to overreact a lot. And a lot of disease, for example, lung disease, when you die, it's not directly because of the pathogen, but because your immune system is overreacting to the pathogen. That's what kills you. And so in colonized animals, the immune system is properly trained and it doesn't overreact to the pathogen. That's why the microbiome has some benefit. And we kind of see that in humans too, like all the diseases associated with immune system, like asthma, allergies, may be rooted in the fact that kind of lost most of our microbiome. And so our immune system is kind of messed up. That's one hypothesis. But it's proven with this with nice experimental setting at least. Okay, so that's the two main benefits. Like disease, pathogen protection against pathogen and digestive abilities. And so the next question is that, okay, so the gut microbiome provides these benefits, but who are these gut microbes? Like, where do they come from? Who are they? And to study that, that'll be the only methodological slide, like wet lab methodological slide of the talk. To study who are they, we usually use metabarcoding. So I don't know if you are used to metabarcoding approach, but basically we extract DNA from usually feces. So extract microbial DNA from the feces of the host, then to do some sort of PCR to amplify a particular region of the microbial genome, and sequence it and define microbial species based on that. So we actually use OTU or these kind of things, but I want to just talk about species today. So we use that methodology to basically list all the bacterial species that we find in the gut. And when you do that, you can actually ask who are those microbes living in the gut of mammals. And the first thing is to ask is, oh, are those microbes the same as the one we find in soil or in the water or in the seawater? And as you may expect, they are totally different. And that's a figure that shows that. So it's a PCA plot of microbial composition. So each point is a sample, a microbiome. And the distance between the point is proportional to the similarity in terms of composition between two microbiomes. So basically proportional to the beta diversity. Okay, so 2 points that are far apart here represent communities of microbes that are very different in terms of composition, while 2 points that are very close together represent 2 communities that are very similar in terms of taxonomic composition of the microbial species. And what you can see is that the points are colored by type of microbial habitats. And today I'm going to focus on the animal gut, but to put in context, here it's compared to all of the microbial habitats on Earth, and you can see that there's, for example, water in blue, or like plant rhizosphere in green. And what you can see is the animal gut microbiome are fairly distinct from all other microbiome found on Earth. So they're composed of distinct taxa of microbes. Okay, the one that colonize your gut are very different from the microbes that colonize the soil or colonize the seawater. Okay. And indeed, if you look at the bacterial tree of life, that's kind of a summary of the bacterial tree of life. The gut microbes that colonize the gut are only found in certain clade of the microbial tree of life. It's not everyone is not colonizing the gut. It's only 2 specialized. For the majority, it's 2 specialized clade of microbes. Doesn't mean that all the bacteria in this clade colonize the gut. There is also bacteria in here that live in soil, bacteria that live in water, and we are actually finding a lot of evolutionary transition between the gut and other habitat within these 2 clades, more than 1,000 actually. It's a paper entered by a PhD student. So most of the microbes are found here. The colonizing the gut are found here. And now today I want to focus on mammals. Okay, I talked about all the gut microbiome found in all sorts of animals, but what about mammal in particular? And when you do again a PCA plot of the gut microbiome of animals in general, so mammals and birds and fishes, what you see again, if you exclude bats, is that mammals here in purple are again relatively distinct from the other animal in terms of the gut microbiome composition. So the gut microbiome of mammals is fairly distinct from any other gut microbiome found in other animals on Earth, except for bats. I cannot talk about bats later. So again, what we see is that the gut microbiome of mammal is rather specific to that clade, or you can call it endemic in a way to mammals. Okay, so I'll use specific or endemic as a kind of synonym way of saying it's a particular set of microbes that colonize mammalian gut. And then you can zoom in again and ask whether the gut microbes are actually specific to species of mammals. So those different species of mammal actually have different gut microbiome. And we actually even found that it's more than that. So if you are here, different species of mammal, what we observe is that for a given microbial clade, this is not all the bacteria we find in the gut, but one particular clade, we actually find that different bacteria colonize different species. So you see, for example, this clade here is only colonizing this host, while this one is only colonizing this host. But even more than that, we find that there is actually a kind of similarity between the phylogenetic tree of both partners, the gut microbe and the host, which means that this specificity is probably very old, and that mammal evolved with their gut microbiome since a long time. Okay, and so why does that matter, that specificity thing I'm talking about since 5 minutes? Why do we care about the fact that, oh, the mammals have a very specific microbiome that is proper to one particular host species? Well, this is because when you try to colonize a mouse with a human gut microbe, people can do that in an experimental system. When you do that, so you have a germ-free mice, and instead of having like mouse microbiome or no microbiome, you put like human microbe in them that actually are able to survive in the gut of Florent. But what you see that this microbiome here in red doesn't provide benefits. So it has the humanized rodent has a lot of infection load similar to a germ-free one, which means that its specificity might have actually a beneficial effect. And this is important because it means that to like have benefit, we need to inoculate specific microbiome, and that's the kind of the idea of a field of study in the field, which is about like, rewilding the human microbiome with like native microbe, but also rewilding the rodent microbiome in the microbiome of this lab mouse that have a very weird microbiome because they've been inbred for generation. Okay, so hopefully I convince you that specificity is an interesting subject because it might be important for this beneficial effect. So, how I'm going to structure the talk. So as I mentioned earlier, the first thing is going to be about first trying to quantify whether that specificity that I showed you like briefly in the intro actually is a pattern that is widespread in mammals and prevalent. Is it really what we observe? I mean, I kind of try to hook you with the intro, but I mean, do we actually observe widespread and prevalent specificity in mammals, in the gut microbiome in mammals? And if yes, what are the mechanisms that can explain the emergence and maintenance of that specificity in different species of mammals? That's going to be like part one and part two. Okay. So if we start with the first pattern part, I'll talk first about to what extent we observe like widespread specificity in mammals, and then I'll explain a little bit whether we see variability between microbes and between mammals in terms of specificity. And so to quantify like the magnitude of specificity, we need to define like sort of measure or metric to quantify that specificity. And what we basically do is that we want to test whether here I'll take a simple measure of host species specificity, where if we expect if there is a lot of specificity that microbiome from different species are fairly distinct. So here you've got two rodent species, okay, with in color in blue or in green. And if there is specificity, we expect the PCOA plot, as I showed earlier. We expect to see in the PCOA plot that the sample from the same species are close together in that compositional space, while the sample from the other species are kind of far away and close together in the space. Okay, which basically means that if you compare individual within species, so between two humans, you and me, for example, we should be more similar than if you compare yourself to a rat. Okay, so that's basically what you're testing. You're testing the within species versus the between species similarity in composition. You basically derive, for example, F statistics to quantify the magnitude of that difference. That makes sense? So just quantifying whether it's all overlapping or fairly distinct between species. That's kind of the simplest measure you can imagine for specificity. Like from a biogeographical point of view, if you're more used to that, you can imagine that the color are continents and the animal are like different region within this continent, and we want to know whether continents have different biota formats. So there's endemicity for each continent. But in my case, it's a bit weird because my continent are alive and moving around, but it's kind of the same idea. And so we tested that with a data set from the Earth Microbiome Project. You might have heard of that. It's a project that tried to sample a lot of different microbiome in different habitats, including the gut microbiome. And so they gather, actually it's not me. In this case, I used about 700 samples representing like about 200 species of mammal. And for each species, of course, I have different individual per species. And we delineated like microbial species using a particular region of the genome of the bacteria, which is 16S, but we don't really care about that. And so basically using this data set, I can test whether that specificity is something that is widespread or not. And what we observe, here you have the plot of the similarity in terms of taxonomic composition between individuals. So 1 minus beta diversity, basically, and separated in between species comparison and within species comparison. So what I was saying before, and I will observe the individuals that belong to the same species have a much more similar gut microbiome than individuals from different species. But you should note that there is still a huge amount of variability within species. I mean, the similarity between two individuals within one species is 25%. So there is a huge variability within species. Okay, but it's still more similar than between species. So we do observe a strong species specificity in mammals, like kind of endemicity for each host species. And what you can wonder is that, okay, but does that specificity extend across phylogenetic scale, and in particular, phylogenetic, like the phylogenetic scale of the host? So for example, do we have similar host species or more similar microbiome than distantly related species, host species? Okay, like does like two species of rats are more similar in terms of their gut composition than the species of rat versus a human? Okay, kind of extended that specificity across phylogenetic scale. And so the measure for that is relatively easy. It's, you just plot the dissimilarity in terms of microbial composition between two species against their phylogenetic distance. And you can just like do a mental test to test the correlation. It's, in a way, it's sort of a measuring the phylogenetic signal in microbiome composition. And so the question here is that does that specificity we observe at the species level, the host species level, also extend beyond the species level and across the host phylogeny? And using actually the same data set as before, but in a different paper, we actually show that measure of specificity that people have called phylosymbiosis, which is kind of not really a good name, but that's how it's called. It's actually widespread in mammals. So here you see that correlation between microbiome dissimilarity between species and their divergence time or phylogenetic distance. And you see that there is a positive correlation for most of the group of mammals, except again bats. So bats are kind of weird. Within mammals, they have kind of non-specific microbiome that look like microbiome of birds. But except from bats, there is a positive and significant correlation between the dissimilarity in microbiome and the phylogenetic distance, so that specificity extends in deep time. And the last measure is the one I presented in the intro. So here I just mentioned the phylogeny of the host, but we can also incorporate the phylogeny of the microbe and measure to what extent there is a similarity between the shape of the phylogenetic tree of the host and of the microbe. It's something that is fairly used in parasitology, and they call that co-phylogeny or phylogeny mirror or congruence of phylogeny, and kind of show that there might have been co-speciation event between the host and the microbe. Not necessarily, but that might be an indicator of co-speciation between the host and the microbe, which means that they've been evolving together for millions of years, and when the host speciate, then the host specific bacteria in the gut also speciate. And so I've shown you one example that is probably the best example we had. A nice, very congruent phylogeny. But we show that what we observe for a particular clade of bacteria is actually widespread across the bacterial phylogeny. With most of the bacterial clade we studied actually show significant congruence with the host phylogeny, more than 50% here in blue. So the conclusion from these three kind of measure of specificity is that it looks like specificity is widespread and old in mammals. So it extends to different phylogenetic scale. So that's kind of what I'm showing you. So just to recall, every mammalian species has kind of a unique endemic microbiome that is just only found in that species, okay, and differ from other mammalian species. And that's particularly true in mammal and particularly strong in mammal. Yeah.
Just a technical question. Yeah. The sample that you presented just before, this includes individuals from the wild or from zoos?
That's a very good question. So it's a mix actually. In this particular paper, it's mostly captive animal actually, but in this one, it's a mix of captive and wild animal. And so there is conflicting result about how the specificity vary. Like, is it stronger in captive animal or weaker in captive animal? It seems that it's weaker in captive animal compared to wild animal. It holds in both cases, but it seems weaker in captive animal, but it still exists. Especially people have been like trying to, they've been like American people working on like Pteromiscus rodents. They have been like having multiple species of captive rodents with the same, like fed on the same thing. And they show that they still maintain a specific microbiome, even though they are fed with the same thing in captivity. It's pretty convincing that that specificity is not just probably not just diet effect, but probably diet plays a role. I'll talk about it a bit later. But I have to say that there is conflicting results about, and I mean, I just saw a paper 2 days ago trying to measure this on captive animals. So when it comes to like this type of measure, which are like not very strong specificity compared to that. I mean, this is very strong specificity at like, because it implies like long phylogenetic, like long time scale of association. When people measure specificity on that on captive animal, they find specificity, but I just read a paper 2 days ago that showed that these results might not always hold with captive animals, like that very strong old specificity. But I mean, we need to stay tuned because it's changing pretty rapidly as we basically like discovering, like we just documenting that specificity, like it's just the early days. So it's changing a lot. It's not very stabilized yet. And so I show that that specificity widespread, but I haven't really talked about how variable it is. So I show that it's variable between mammals. So for example, bats seem to have not a specific microbiome, but I haven't really talked about how is it variable between microbes. And we expect that some microbes are going to be very specific or specialist, and other microbes are going to be more like generalists and kind of be able to colonize different species. And that's what we showed here. So again, I mean, this is just one example of a specificity measure. And when you basically try to measure that for all the clades, you see that depending on the clade, you've got wide variation of what we call a co-speciation pattern, or like specificity in a way. So very high variation between like very specific or like specialist microbes to very generalist microbes. So there's a huge amount of variability actually within the gut microbiome. And I'll use that later to kind of try to test some processes about what can shape specificity. So keep that in mind. I also measure that with a simple measure of what we call host range. So it's like for a given microbe, like how many hosts does it colonize as a measure of like being a generalist or a specialist. And what you see is that, so here is the specificity of one given microbial species. So basically the number of hosts colonized and like standardized measure. And what you see there, so it's not really, as you get down here, it's more specific. And this line here is kind of the significance threshold. So you see that most microbes are actually specific, more than, I mean, significantly specific, but there is a huge variability between like no specific microbe to highly specific microbe. And the key question would be to explain why some microbes are super specific while others are actually not specific. Okay, so just to summarize that first part, which is like very pattern oriented, but it's kind of necessary in this field because we are just starting to describe the document pattern. I've shown that there is strong specificity in mammal for the gut microbiome, and that there is a high variability between mammals and also between microbes. And so now, I mean, the most interesting part is trying to understand what are the causes and the mechanism of this specificity. What can explain the evolution and maintenance of that specificity over generation. And so that's the most interesting part, but it is also the trickiest part, because the gut microbiome is a weird system. Because it's in a way it's similar to like classical community ecology things, because we have like an ecosystem of microbes and we're trying to understand the composition of these communities. But it's different from community ecology because our like the support is not just space, but it's a host. It's something that is living, that is moving around, that is evolving. So it's like a community within an animal. It's fairly different than just a community of plants on one thing side. So it's similar to community ecology, but different, also more complicated. And it's also relatively similar to like the parasitology and the symbiosis field of research, because as I said, those microbes provide benefits to their host. But again, it's fairly different from those fields because in this field, people usually focus on like parasite or symbiont. And when they focus only on one parasite, or one symbiont. Classical textbook example in parasitology or symbiosis, it's about like one symbiont. I don't know, the Buchner and aphids symbiosis, it's like one symbiont, one host. And all the theory, everything has been developed for this kind of relatively simpler system, where you have so one symbiont, one host, and there's usually a very tight and obligate relationship between the two. While in this case, well, it's a community of like hundreds of species, so hundreds of symbiont, one host, and this relationship is not very strong. I mean, there is specificity, but it's not an obligate relationship. As I say, when you remove the microbe, the host doesn't die, right? It's not an obligate relationship. So it's interesting, but it's a weird system because you cannot really apply things from here and not really things from here. You have to try to make a mix. And that's what I try to do. And we can maybe take one angle, the angle of the community ecology framework. I guess maybe you know that more than the parasitology thing. And people try to understand what are the processes that shape the composition of the community, which is why I'm interested also. They usually, we used to use the filtering framework, right? Where you've got a species pool. So here in my case, a microbial species pool of like adapted microbes that so they can live in the gut, or more or less live in the gut, and then they can colonize the gut of different host species based on three main processes. What is called filtering, so which is basically the fundamental niche of this microbial species. So are they actually able to live in that particular gut? It can be explained by some sort of dispersal limitation. So like maybe you are not present in a given community because you were not able to reach it. And finally, it can be also explained by microbial interaction. So for example, competition. And so that are the three main type of processes that have been used to explain the pattern of community composition. And so I'm going to try to basically use this framework to explain some of the pattern we see with specificity. And I'll focus today on the filtering in yellow and the dispersal in gray only. And so when you think about that filtering, what we call in community ecology, the environmental filtering effect. In this case, it's more like a host filtering, so the host is actually filtering the microbes. And in this field, it comes in like two flavors that are actually fundamentally different. The first one, which is the one you most commonly see, and it's usually the most exciting and what people usually think when they think about host filtering is what has been called the holobiont or hologenome theory. I don't know if any have heard of that. Have you heard about the holobiont and the hologenome theory of evolution? No. So it's basically a theory that say that we shouldn't, when we try to understand the evolution of animal or plant, we cannot just think about the plant itself, or the animal itself. We need to include all those microbes that come with the animal or the plant, and we need to think about it as a holobiont. So it's not just an animal or a plant, but it's a mix of a plant or an animal with all these microbes that host a hologenome, which is the genome of the host and the genome of the microbes. And it kind of makes the hypothesis that there is a coevolutionary dynamics, like for example, the red queen dynamics between the host and the microbe, and that at the end, this holobiont, so the host and its microbes, form like a coherent evolutionary unit that evolves in time. And that has been used a lot to explain that specificity. Because if this is happening, you really have a coevolution between the host and its microbes, you expect that over time, specificity is going to emerge. But I think this is it's a very complicated theory, and I think there is a simpler way to explain the specificity pattern I observe, and I called it the byproduct theory. So the fact that there might be filtering by the host, and in this case, the filtering is kind of an active process by the host, kind of choosing the good end, like they're choosing the good microbes and kind of removing the bad microbes, like it was like the result of an adaptation to choose the good microbes in purpose to provide benefits. And that's what kind of the whole biome theory says, and that the sort of filtering they're thinking about. In this case, it's much more simpler. It's just that different guts of different animal species are physically and chemically different. And that might just filter out the microbe without necessarily being for the purpose of filtering the microbe. It might be just a byproduct of something else. And I mean, the best example of the host trait that would filter microbe from a species pool without having evolved to filter those microbes is typically diet. So different species are going to consume different diet, and that's probably going to filter out different set of microbes. And you will probably observe specificity. You could observe specificity just because of that. And the diet, I mean, the evolution of a particular diet didn't evolve to filter out the microbes to be beneficial. It's more like the diet evolved in the first place, and then just the microbe adapted to this diet as a byproduct. So that's just one theory, right? One idea. And what I first did is to try to check whether this byproduct theory can be plausible. Can you explain the specificity we observe in nature, but just from a theoretical point of view? And to do that, I just conducted a simulation where basically you start with assembling microbial community with given processes. So in this case, filtering by a host trait, but I didn't evolve the host trait to filter the microbe. I just assigned a trait to a host that would filter the microbes. And for that, I used actually a software that has been developed here by Lorgalien when she was here in 2015, and it's called VirtualCom. And I varied the phylogenetic signal of that host trait that filter out the microbe. And then in this simulation, I measured the specificity I could observe based on this byproduct filtering process only. So no coevolution, no holobi, anything. And then you can basically conclude whether that simple process can produce significant specificity pattern, and to what extent it matches what we observe in reality. And what I showed that indeed, when you have a host trait, so for example, diet, that has a very strong phylogenetic signal, so closely related species have a similar diet, and the microbial filter out by this trait, you observe here a high chance of observing specificity. So here measured by the phylosymbiosis measured specificity. So meaning that just with this simple process, and if you have like a phylogenetic signal of the host trait, you can produce the specificity pattern we observe in nature without relying on coevolutionary dynamics. And interestingly, when you compare the kind of the expected magnitude of specificity here with measured with a mental test here, and when you compare it with what can be observed in nature, they are within the same range of magnitude. It doesn't necessarily mean that the theory is right, just mean that the theory could explain the specificity we observe in nature. And so, well, next question is to test it. It's nice to do simulation, but you actually want to test it. And it's actually work in progress. So the idea here is to sample at the same time the microbiome of different species to measure that specificity and also measure the diet of each of these individuals and try to find whether the specificity we observe is correlated with the diet. And so I've been doing that all summer in Switzerland and basically sampling rodent poo all over Switzerland. Actually also nearby, I mean, mostly nearby Zurich in Pizzol, where I had collaborated trapping rodents, so I took advantage of that. So stay tuned to see whether the diet can explain that specificity pattern. And so the last part is about dispersal and to what extent dispersal limitation of microbe can explain the specificity pattern. Again, it's it's a process that do not rely on coevolution, and that kind of is an alternative to the holobiont theory. I'm going to be quick here, but basically the idea is to what extent dispersal limitation between host species can basically foster unique microbiome found in the blue species versus the green species. And for a long time, I mean, dispersal limitation in bacteria was a joke, and people would make fun of it. People would think that there is no dispersal limitation in bacteria, and it's kind of captured by this famous sentence, at least for microbiologists, that everything is everywhere but the environment select. Saying that all the microbes are everywhere, they can disperse everywhere, but then they are adapted to different environment. That's why we see biogeographic patterns. There is no dispersal limitation in bacteria. And so this has been challenged for gut microbes, particularly. In particular, within host species, we observe that the social network between individuals predicts very well the gut microbiome composition. So individuals, so it's been shown here in primates. So individuals that interact a lot socially, that touch each other a lot, in this case, grooming, they actually also have a more similar microbiome because they probably exchange a lot of microbes when they touch each other. It seems that there is some potential dispersal limitation that act on the composition of the gut microbiome. They have been shown in primates, but also in rodents, also in humans. So dispersal limitation might exist for gut microbiome. The question here is more about how does dispersion happen between generations. Like here, I was talking about just individual within a population, but to really explain the specificity we observe between species, we need to think about how the dispersal limitation acts between generations of the same species. And to understand that, I just need to know that how you transmit the microbiome to the next generation. And what you need to know basically is when a newborn is sterile, so there's no gut microbe in the gut of the newborn, and basically the gut microbiome developed over a couple of years. And it's actually an ecological situation. You have a high turnover for the first couple of years, and then it's kind of stabilized. So the microbiome kind of established during the first years, and the question is from where? And there is a very elegant study showing that it's actually from the mother, because again, usually in close contact in mammals. And the study is very elegant because what you do is that you do some cross-postering experiments. So you take, let's say, two lineages of a given lab mice, in green and blue, with the newborn, and you basically exchange the newborn between the mothers. And so you've got all the case, like the normal case here, and then a kind of mixed up case. And if it's actually the nursing mother that is the source of the microbe for the newborn, then you expect that the microbiome of the newborn will resemble the nursing mother and not the real mother. And that's actually what we observe. So the microbiome cluster according to the nursing mother, which means that the source of microbes are actually the people that are around you during the first years of your life. And if you extend that to like between species comparison, you can easily imagine an ancestor species in yellow with a given geographic distribution that's probably going to speciate because of allopatry in general in mammals. And that's probably going to reduce dispersal limitation between individuals of the two species just because they don't see each other often. And that probably can explain some of the specificity we observe, just that simple process. And to test it, and that's kind of the last part here, a way to test that, whether dispersal limitation is actually important for specificity, we can take advantage of the fact that there is good and bad dispersal in microbes. So microbes tend to be can easily switch between guts because of some traits and other not. And if dispersal limitation is important, we expect the good disperser to have like low specificity because they can basically easily switch between individuals of the same or different species, while bad disperser will probably have high specificity in theory. And for microbes, a good disperser is a microbe that is, or gut microbe, is a microbe that is able to live outside of the gut for a while. So either by producing spores and of resisting the outside environment, or by being tolerant to oxygen, because most of the gut environment is mostly anaerobic, there is no oxygen, and most of the gut microbes are actually very sensitive to oxygen. And so the ones that are less sensitive to oxygen, they're probably it's probably easier for them to live outside of the gut, for example, in this table for a while, and enabling to switch between hosts. And so what I did, I mean, what is interesting is that we observe that within species, within human, for example, the bad disperser actually less widespread in human population than the good disperser. And it's measured its prevalence, but it's basically meaning that the good disperser are like more widespread in human than the bad disperser. So it's encouraging, but does it also explain the specificity between species that I showed earlier? And to do that, I again use the same data set, the Earth Microbiome Project, quantify the specificity of each microbes here with the host range, and also kind of look into the literature to establish whether I was a good or bad disperser, looking at sporulation abilities and oxygen tolerance. And what you see here will be the specificity of the gut microbial species, if it's an anaerobe or so basically can tolerate oxygen or can tolerate oxygen, and here a microbe that's not forming spore or can form spore. So good disperser, bad disperser. And what we observe for oxygen tolerance is that the one that are very sensitive to oxygen, so cannot really live outside of the gut, they are more specific than the one that can actually live outside of the gut. So kind of confirming our initial hypothesis. But we don't see any trend for sporulation ability to set. But that's using one particular data set. If I use another data set that is more resolved, like have more resolution on the microbial species, actually see again an effect of the oxygen tolerance, but also a slight effect of sporulation ability. So microbes that are able to form spore, they are actually less specific than the other one. So maybe there is a trend there. So in conclusion, the specificity I observed seems linked to oxygen requirement and to extend sporulation ability, which might be due to an effect of this dispersal limitation. So, I mean, this is not totally conclusive, but this suggests that dispersal limitation can be one factor that explains the specificity we observe in mammal, and so we don't necessarily need to rely on complex holobiont or genome theory of coevolution. Simple processes as dispersal limitation can actually explain part of this pattern. That's what is on the conclusion here. I observe a strong specificity in mammals, and usually people use that as an argument to say, oh, we evolved for a long time with our gut microbiome, look how specific it is. But what I showed is that actually alternative models that don't rely on coevolution can also probably explain most of this specificity observed in mammals. I think that's it. I'm just going to acknowledge some funding and some colleagues. So we had, as Rafi said, at UNIL, but also at UBC, where I carried most of this work, and also other colleagues, especially from the Earth Microbiome Project. And I'll thank you for your attention. Happy to take any questions.